So now we're going to focus in on how we get from DNA to RNA, looking at the different enzymes and regions that are involved in this process of transcription. So there is a specific enzyme, and unlike DNA replication, this enzyme is able to do pretty much the whole thing with just a little bit of help from some outside stuff. So we've got RNA polymerase, and that sounds a lot like DNA polymerase because it does similar things. What it's going to do is it's actually going to be able to bind here to a start point. Start points are typically called a promoter sequence. This is going to tell the RNA polymerase this is the place where the gene starts. This is what we want to transcribe. And so we're going to start at this promoter. RNA polymerase does not need a primer. What it's going to do is it's going ahead and it's actually going to be able to open up the DNA on its own and start adding molecules of RNA. Again, it's going to build in the 5' to 3' direction. And as it goes along, it's going to use the template strand. So in this case, the bottom strand is the template strand. And so you can see how everywhere RNA polymerase goes, it's complementary base pairing to the bottom, the template strand. And it's going to do this all the way down the DNA molecule until it reaches some place that says this is the end. So this is typically called a terminator. So a terminator is a region of the DNA that says this is the end of the gene, RNA polymerase can stop now. So what we end up doing is we make a short piece of RNA that is called the transcript. So in a prokaryote, this would be the mRNA that's ready to go. In a eukaryote, this is called pre-mRNA because it still needs to be processed. The region of the DNA that was transcribed, so this region on the DNA, is the transcription unit. From the promoter to the terminator, the promoter is the start and the terminator is the end of that transcription unit. Transcription is going to happen in 3 steps. The first step here is called initiation. So initiation means to begin something. So the first thing that's going to happen is the promoter is going to signal transcription start point. So we have a section here, which is the promoter. In a eukaryote, there is typically a region in here called the TATA box. So the TATA box is just a region with many T's and A's, and you can see there's lots of T's and A's that are repeated, and that is a region inside of the promoter. So remember, the promoter is a piece of DNA that says start here. The next thing that's going to happen is some proteins called transcription factors are going to help bind here. Without the transcription factors, the RNA polymerase doesn't really know where to go or when to start. So the promoter really tells the RNA polymerase this is the beginning, and then as soon as the transcription factors bind, they say, okay, this is now the time to start at this beginning and go ahead and transcribe this DNA. So that creates what's called the transcription initiation complex. Once we have the transcription factors bound to the promoter, and then we have our RNA polymerase bound to that promoter as well, we are ready to go ahead and get started. At this point, initiation has begun. We are going to lay down the first pieces of RNA, again in the 5' to 3' direction, and then we're going to move on from initiation to elongation. The second step in transcription is elongation. This is talking about elongation of the RNA. So this is going to happen as the RNA polymerase is going to move along the template strand. You can see that the RNA polymerase is going to untwist the double helix on its own, and it just does this in a short region, about 10 to 20 bases at a time. So you can see this region it's already come past, and as it's passed, it has rejoined back into a double helix. This region over here is downstream, so once the RNA polymerase gets over here, it will untwist that small section of DNA. This relieves the need for something like helicase that opens up DNA. It also relieves the need for having to kind of hold the DNA open or cut to untangle the DNA. It's not going to get tangled because we're just opening a small portion and then we're letting it close back up again. And so again, you can see here that it's only adding complementary base pairs to the template strand. So whenever it sees a T, it adds an A. Whenever it sees an A, it adds a U. Wherever it read a G, it added a C. And if there had been a C, it would have added a G. So here's a C, it'll add a G there. And so these are going to end up being the RNA that we're going to use to make our protein in translation. The third step of transcription is called termination. So terminating something is ending something. They're a little bit different in a bacteria versus a eukaryote. So in bacteria, what's going to happen is it's going to reach a sequence called a terminator, and once the RNA polymerase gets to the terminator, it will stop transcription. The terminator says, this is the end of the gene, go ahead and stop here, and it won't do any other further modifications. Eukaryotes are going to have to further modify the RNA. So what happens is the signal that the gene is over is going to transcribe off of the DNA something called the polyadenylation signal sequence. This is going to be a signal that says this is where we're going to add a lot of adenines. So it is a poly, many. Adenylation means adenine. Signal sequence. So that's going to signal to add many adenines. After this, it's going to be released several nucleotides later, and then the eukaryotic mRNA will go on to be processed. So at this point, transcription is done. We have copied the section of the DNA that we wanted to copy, called the template, and it is now in the form of RNA. If you are a bacteria or a eukaryote, then you're done. Your messenger RNA is finished, and you can go ahead to translation. If you're not a bacteria, you're a eukaryote, we're going to go through a process called RNA processing. RNA processing is going to take the pre-mRNA and turn it into the mature mRNA, and it's going to make 3 changes. The first change is that the 5' end is going to receive a 5' guanine cap. So here is the pre-mRNA, and you can see this is the start of the pre-mRNA. This is the end of the pre-mRNA. And the first step we're going to do is we're going to add a 5' cap. We're also going to cap the other end. So the second change that's going to occur during RNA processing is adding a poly-A tail. Again, this just means many adenine tail. So that's going to happen on the 3' end. So here's the poly-A tail. Notice this happens right after the poly-A signal. So the poly-A signal, which stopped translation, is right before the poly-A tail. That is helpful in telling the adenylines where they need to go when we add this 3' cap on there. Adding caps to the ends is going to be extremely useful for eukaryotic mRNA. They're going to help the mRNA get from the nucleus out to the cytoplasm. In the cytoplasm, there are enzymes that break down RNA. So by having a protection on both ends, it's going to help prevent that and slow that down. It also helps the ribosome to attach. So when the ribosome finally finds this RNA, it's going to be able to use that 5' cap to help it attach to the correct end so that it can read it from 5' to 3'. So you can see that the actual gene that we copied is going to be here to here. The third step in RNA processing is called splicing. So splicing is actually going to take some regions of the mRNA and remove them. So there are some non-coding regions that we call intervening for intron. So intervening, intron. These do not get expressed. These get removed. The other regions are called exons, EX for exons, because they get EX expressed. These are going to have to stay because these are what actually code for the protein. The introns do not code for the protein, so they need to be taken out. So splicing is going to remove these introns. These need to be cut away. So we do this in something called a spliceosome. So a spliceosome is a variety of hydrolytic enzymes that exist together, made out of proteins and some other small RNAs. Those are going to bind to the ends of the intron, and these act kind of like molecular scissors. So they're going to cut that end and cut that end, and then remove the intron, and then at the same time they're going to stitch these exons back together. So what we end up doing is we end up removing these sequences that don't code for our protein, and we end up with a much shorter mRNA. So this is our mature mRNA. It is finished. It has a 5' cap, a 3' tail, and we have removed the introns. So 3 steps in RNA processing. We think that some introns may actually help to regulate gene expression. There is an idea called alternative RNA splicing. So here you can see a pre-mRNA. It's got exon 1, exon 2, exon 3. Those are the regions that are going to get expressed and then eventually make up this protein. So here's domain 1 of the protein that was coded from exon 1. Here's domain 2 that was coded from exon 2. Here's domain 3 that was coded from exon 3. We're going to remove these introns. There is an idea that we could actually use one gene, so this is the gene, the DNA, to make different proteins by removing different exons. So if you removed this exon, this domain would no longer exist. We would actually get a completely different structure of a protein. A lot of the time, if you remove a whole exon and you change the shape of a protein, it's going to change its function and not work. However, this exon shuffling might be a way for evolution to produce brand new proteins that have never been seen before by using different combinations of exons within a gene. So this may actually benefit the overall variety and diversity found in organisms.